Going live. Great.
Okay, we're good to go?
We are.
All right, so I think there's still some people joining in, but we'll get started. My name is Catherine Uhazy. I'm a new assistant professor of ophthalmology at the University of Pennsylvania, and we're delighted to welcome you today to today's Vision Science seminar series featuring Dr. Tom Ray, who's a professor of biologic structure at the University of Washington. Dr. Ray's lab studies retinal development and retinal regeneration with a particular focus on Mueller glial cells and their regenerative capacity to form neurons and restore function to retinal neurons. He's received numerous awards, has published many landmark papers in the field, including the first evidence that mammalian Mueller cells can actually, with the help of a proneural transcription factor, ASCL1, can actually form functional interneurons in the mouse that can connect with existing circuitry and actually respond to light. His work is inspiring not only to the millions of patients with vision loss due to retinal cell death, but also on a personal note to young investigators like myself who have followed your work and are just starting out in this field and have really been inspired by your findings and become hopeful that there will be soon better treatments for vision loss in the near future. So without further ado, it's my pleasure to welcome you, Dr. Ray. Thank you for joining our seminar series today. And we really look forward to hearing some updates on these exciting projects.
Great, thank you for having me. And I do want to say that, I know all the great work that's going on at UPenn in vision science and in prevention of degeneration and slowing down retinal diseases and hopefully treating them so we don't lose neurons in the future from degeneration. But I see my approach here, the thing I'm going to talk to you about today, is more of like plan B in case we don't actually get to stop all the degeneration from some of these other diseases. What can we do to actually put neurons back? And I'm going to tell you about our approach of repurposing glia to regenerate neurons. And I'll hopefully convince you that while it's still early days in this approach, that there does seem to be some traction here that we're getting, and I think that there's some real possibilities that someday this will be in patients. So hopefully we're somewhere along the trajectory towards those goals. I do have a couple disclosures. Some of the research in my lab is funded by Genentech. Some of the research in regeneration that I'll be talking about today is funded by the Genentech Corporation. And some of the work on reprogramming transcription factors has also been patented by the UW. So currently it's not licensed, or I'm not working with any company to develop this, but I hope someday that we will. All right, I know I don't need to really say much about the retina and the background here, but I'll just say a few words that for those of you who aren't in visual sciences, the retina is the place where light energy gets transduced into electrical potential changes that allows us to see. And it's a part of the brain. Essentially, it's an extension of the brain, if you will, during development. And this part of the brain, then like other parts of the brain, doesn't repair itself after injury very well. So in general, neurons don't get replaced after they're lost, and axons don't refine their targets and reconnect after they're lost. Long axons don't reconnect with their targets very effectively after they're lost. And this is characteristic of the entire central nervous system, including the brain and spinal cord and the retina and optic nerve. And so people have struggled over the years, and it really worked hard to try and find ways to repair these tissues. And there's lots of approaches being taken right now, but we're still, I'd say, a ways off. This just shows a summary of some of the diseases that lead to death of retinal neurons. And some of these diseases I'm sure you guys are all aware of, like retinitis pigmentosa and macular degeneration. Macular degeneration results, it's caused in part by the atrophy of the pigmented epithelial cells, which then leads to a loss of the cone photoreceptors, and that leads to a loss in vision. Retinitis pigmentosa, primarily a disease of the rod photoreceptors, but also ultimately can involve cone photoreceptors, and again, serious visual impairment. And then other diseases like glaucoma, where ganglion cells are lost, and retinal arterial occlusion, where inner retina is damaged. And all of these diseases, when, if we could intervene early, either with gene therapy or different types of growth factors or whatever, people have tried over many years to try and figure out ways to stop this loss of these neurons. But when the loss of neurons occurs, there isn't an endogenous source for regenerating them. And so then the gene therapy obviously wouldn't work if you can't treat early enough in the disease. Now, that's not true of some animals. They have this ability to actually regenerate their retina. And amphibians, newts and salamanders, these eurodelian amphibians, are amazing at this process. Here is an example of a fire-bellied newt. And Sanai Sakami in my lab years ago just recapitulated some of the classic work in this area. She removed the retina, and then after a few weeks, 2 weeks, you start to see new unpigmented cells that actually are derived from the pigmented epithelium. And these form a new layer of cells that then goes on to reconstitute the entire retina after just 5 weeks. So pretty impressive ability. This ability to regenerate the entire retina is shared in other non-mammalian vertebrates, but in birds and in fish, the neuroregeneration that does occur actually comes from Mueller glia rather than from the pigmented epithelium. And Andy Fisher in my lab, when he was a postdoc, he and I first discovered this process that occurs in bird retina, where new neurons could be generated. In this case, a neural marker HUCD, which labels amacrine cells. These new neurons were regenerated after NMDA injury in the bird retina. And this was really some of the first evidence that a warm-blooded vertebrate would regenerate, as opposed to fish and frogs. Now, the way that he did these experiments was he injected the neurotoxin NMDA, and then followed up with a EDU or BRDU, in this case, injection. And you can see how the retina was relatively, in fact, very quiescent prior to the damage. But after this injection of NMDA, there's a large number of cells that re-enter the mitotic cell cycle. And these cells undergo one round of cell division, and then they turn on proneural genes. And in this case, one of these genes, ASCL1, was one that we had actually originally identified in the chick embryo and were the first to clone it. And so this gene is a proneural transcription factor that gets turned on in these mitotically active cells in the retina. And it turned out these cells were Mueller glia. He did lineage tracing to show that the Mueller glia actually are the ones that give rise to these neuroprogenitors. And within a few days of injury, the bird retina spontaneously increases in its expression of these progenitor genes, followed by an increase in more definitive neural markers, and ultimately the generation of neurons. So this is a spontaneous process that occurs just by the injury, the NMDA injury. And that same thing is true in fish. David Hyde's group and Pamela Raymond, Peter Hitchcock, a number of, Dan Goldman, in fact, is probably another one of the seminal people in this field, where they also showed that Mueller glia were the source of new regeneration in the fish retina. And so after our paper came out, Andy Fisher and my paper came out saying that Mueller glia were the source in birds, it had been known that fish regenerated the retina, but it wasn't really, no one was really sure from what cell type it came. And Pam Raymond had suggested Mueller glia were the source, but in fact, there were a number of possible sources. And in fact, Mueller glia weren't the number one pick of anybody. But Dan actually showed that fish regeneration comes from Mueller glia as well. And so this just shows a paper from Ryan Thummel and David Hyde's group, that after rods are damaged in the fish and completely wiped out by intense light damage, the rods are replaced over time. And it really just takes a couple weeks for these rods to all get put back in the fish retina. And it's the Mueller glial cell that re-enters the cell cycle, shown here by PCNA staining, and that these cells then will go ahead and repopulate the damaged rods and or any other cell type that gets injured in the fish. So the fish is basically able to reconstitute all of its different retinal cell types through this regeneration process, but it's through the Mueller glia and their cell divisions that allows it to do it. So this just summarizes some of the work. This was from a review that Mike Carl and Deepak Lampa and myself wrote back in 2008. But the idea is that there's a non-neural cell in both the Urodale amphibians and in these other vertebrates. These non-neural cells retain the ability to undergo proliferation into adulthood, whereas neurons do not. And so these non-neural cells can be spontaneously triggered to re-enter the cell cycle and reproduce neurons after injury. And because it's a spontaneous process, nothing really was needed to trigger this. It occurs just naturally in these guys and in fish. Now in birds, it does occur naturally, and these cells do re-enter the cell cycle and go ahead and make these different types of neurons, but they don't make all types of neurons. We never saw in the chick photoreceptors, for example, made by the Mueller glia. And so that always puzzled us, like, why is the bird limited in terms of its repertoire of what it can give rise to? And I would say we still don't know the answer to that. We haven't studied chick for many years, but Andy Fisher continues to study them, and it's not clear what is restricting the more totipotency of retinal production, cell production, to this more limited multipotency that you see in the bird. But in mice, really, there's very little spontaneous reentry into the cell cycle of the Mueller glia. As far as we can tell, Mike Carl looked pretty hard for this and really found very little spontaneous reentry into the cell cycle after Mueller glia, or after there's an injury to the retina, either NMDA or from a photoreceptor degeneration. So mammalian regeneration seems to be quite limited in its intrinsic ability. Now, back then in 2008, we thought about this a little bit and framed the problem the following way. Well, normally during development, a progenitor cell is able to give rise to both neurons and Mueller glia. And it was really some of the early work of Connie Stepko and Bill Harris and others that showed that if you labeled a single progenitor cell during retinal development, that cell would go ahead and make a clone that contained both neurons and glia. And this is now a pretty well accepted principle that there are multipotent progenitors that have potential to make both neurons and glia. And so we wondered what's really different between the glia that gets generated and the progenitor cell, because this glia sticks around your whole life, and in fish it's able to, revert back to a progenitor-like state. In mammals, it seems to be a one-way street. What changes as these progenitor cells become Mueller glia? And so we had a mouse line that actually had expressed GFP in both progenitor cells, shown here in the retinal section of postnatal day 3. And all these labeled cells are retinal progenitors, and they're still actively undergoing mitosis and making new retinal neurons. But then quickly by postnatal day 7, and definitively by postnatal day 10, there are no more progenitors left in the retina. Instead, what you see is this same GFP, Has5 GFP, is expressed just in the Mueller glia. And that continues to be true throughout the animal's life. So we've FACS purified these cells from these different stages of development and then put them through cDNA microarrays to try and find out what transcription factors are more highly expressed at this stage and are lost by the time the cells get to this stage. And so these are the transcription factors that we found. You can see they're highly expressed as shown by this red labeling here. And then as the expression gets lower and lower, you can see that reflected in the cells, because these bars becoming green. And so, for example, ASCL1, which I told you about in the bird, that's important in its regeneration. ASCL1 is high at postnatal day 0, but it's gone in the P21 Mueller glia. And this was basically our list that we prepared. Now, at that time, there had been the seminal work already of reprogramming fibroblasts into iPSCs, and the idea that you could use a group of transcription factors to reprogram to another state was really something that we thought made a lot of sense in this context. That essentially what was going on in regeneration is that there's kind of a regulated endogenous reprogramming that's going on. And that in the transition from a Mueller glia to a progenitor, it's quite likely, even in fish, that it's the transcription factors that are really driving the process. And in fact, so we thought maybe we could use a subset of these transcription factors in reprogramming Mueller glia into neurons in the mouse. And so we set about testing that. One of the ones that was our favorite right from the get-go is ASCL1. And as I said, we were the first to clone this in the bird back when people were still cloning genes. So we called it CASH at that time. But it's been renamed ASCL1 for all species to have the same. That people got tired of CASH and MASH and HASH and all the various species-specific names. So it's now called ASCL1, but it's the same gene. And we have found, like I said, that this gets upregulated in the bird. Dan Goldman's group found that it's necessary for regeneration in the fish, so that if they actually knock down ASCL1, with a morpholino, they ended up repressing the regeneration, even the re-entry into cell cycle that occurs after the injury went from lots of labeled cells here to practically none. So we thought that was a good choice, but we really cloned all of these into lentiviral vectors to express all of them. And we had various combinations of these things that we would put in and then test to see what kinds of effects that would have on the glia. Julia Pollack really spearheaded this work as a graduate student. She really was one of the first to develop the techniques to culture mouse Mueller glia. It had been done by Vijay Sarthi here at the University of Washington back in the 70s and 80s, I guess 80s. And he had not done this with mice, though. He used rats. And in fact, rat Mueller glia grow really, really well in culture, but mice not so much. So Julia really had to do a lot of fiddling to finally get these guys to grow well in culture, and then thoroughly characterized that they were actually Mueller glia by using things like our HES5 GFP mice, and also a variety of different markers. And they grow as flat cells, sort of fibroblasty-like cells, and look a lot like astrocytes in culture when you grow them, but you can distinguish them from astrocytes by a variety of different markers. And so she grew these cells in culture and then started adding transcription factors back to these cultures. And ASCL1 produced the most profound effect right from the start. You could see that instead of these flat cells that you see here, now you see these neuronal-like cells popping up all over the cultures, and they make long processes. And it was really pretty obvious. Within 4 to 6 days, you see this happening. None of the other transcription factors we tried had quite this strong an effect and quite this dramatic an effect. But on the other hand, we have done some combinations more recently, and we find that I'll get to that at the end of the talk, that there are additional transcription factors from that early list that we're testing now that we see additional effects. And Julia just did a lineage tracing experiment just to verify these came from the glia using an RLBP Cree ERT2 TD tomato mouse. And this mouse line was generated by Ed Levine. He was at Utah at the time, now at Vanderbilt. And Ed really, this reagent is a great one because the RLBP is restricted to Mueller glia and pigment epithelium in its expression. So Julia was able to activate that Cree with tamoxifen, and then that recombines the floxed stop in the TD tomato, so we could track all the cells, tomato-derived glial cells. And then she was able to show that after she treated with ASCL1, she could get these neurons, and they would be both red and green. So we knew that we could make these things in culture by overexpressing these transcription factors. We could make neuronal-like cells, and we characterized those. I don't have time to really get into details about it, but we saw some changes in their electrophysiology consistent with neuronal differentiation. We also did microarrays on the studies, and we sort of thought, we don't know exactly what kind of retinal neuron they are, but they have the most likely characteristics in culture with bipolar cells. And so Julia was pretty convinced she was making bipolar cells in culture. But in culture, it's really hard to tell that you've really got a bipolar cell. And at the time, we didn't have as many genes for bipolar cells as we do today. So Yumi Ueki postdoc in the lab generated a TETO ASCL1 mouse that allowed her to overexpress ASCL1 in Mueller glia. And we both made one of these in-house, and Masao Nakafuku in Ohio was also making one. And so we swapped mice, and his is actually green, which we ended up finding to be more useful for a lot of ours. And ours drove ASCL1 TD tomato. So ours was red, but it was nuclear. So we kind of liked his green one, because what it does is it makes the glia green in their entire morphology. So you can really see and track the beautiful morphology of the green cells. So we're looking at the retina of a mouse that has ASCL1 driven from a TETO promoter, and also GFP downstream from that. And then we cross it onto a mouse that will allow Mueller glial specific expression of the TTA to drive the TETO, and that using either the RLBP or another glial specific promoter mouse, or a Cree mouse called the GLAST Cree, for the GLAST gene that is also glial specific. So when we look at these mice, we see nice labeling of the Mueller glia, and the GFP is confined to Mueller glia. We never see any neurons under these conditions, under normal conditions. And so Yumi then decided she would try to test whether or not expressing ASCL1 in the Mueller glia would allow them to regenerate. So her first experiments were really done in older mice, but she didn't see much. So we switched to these younger mice, where at postnatal day 14, she would inject the tamoxifen at postnatal day 12, when the glia are already generated during development. They're finished being, they're done for about since postnatal day 8 or 9. And so these are all glia by postnatal day 12 that the transgene comes out in. And then she gave NMDA injections at P14, and then collected a week, either 1 or 2 weeks later. With no injury, the cells look fine. They're expressing ASCL1, but they look just like normal Mueller glia. But with injury, NMDA injury, she found that now the cells started to take on these really distinctive neuronal morphologies. And that's what we see here. This is an amacrine-like cell, and this guy here is a bipolar cell. It looked a little rod-like. But this effect only happened in young mice. So if we instead did the same thing in adult mice, like at postnatal day 30, the glia continued to look just like this. So we realized that there was some promising evidence that these glia could now give rise to neurons in the young mice. And here's a HUCD, which is staining which stains the amacrine cells, just like in the chick, and CABP5, which is staining the bipolar cells. And you can see we get both bipolar cells and amacrine cells in these young mice. But again, didn't happen in the adult. And so we spent some time trying to figure out what else we could do to make adult mice regenerate. And we turned to the possibility that epigenetic factors might be limiting the regeneration. And for this work, Matt Wilkin collaborated with John Stamatoianopoulos, who had just invented this technology for looking at accessible chromatin. This predates the ATAC-seq, and John was using a limited DNA-seq method, where you'd digest the DNA with DNase, and then these little fragments that got released, you could use to prime and then sequence and then show where the chromatin is accessible. And Matt Wilkin in my lab really worked with STAM Lab and generated maps of normal retinal development as well as during regeneration. And what we found in regeneration is or what we found when we compared Mueller glia with the progenitor cells, is that for many genes, the accessibility around those genes, which shows that the cis-regulatory elements are open for regulation on those genes. Genes like neurogenin, the chromatin was actually quite similar between Mueller glia and the postnatal retina. But when we looked at genes like rhodopsin and NR2E3 and NRL, now we see that the glia have no accessibility in these areas, but that the adult retina has plenty of accessibility in these areas. Obviously, the rods are open around rhodopsin and NR2E3 and NRL. So we thought maybe the part of the problem with getting glia to regenerate in adults is that they have lost accessibility near some of these key genes. So Nick Jorstad took on this project of trying to inhibit histone deacetylases. Normally, histone deacetylases are involved in setting up a repressive chromatin, whereas histone acetyltransferases will put an acetyl group on H3K27 and open up the chromatin so that it can be activated by transcription factors. And so we thought maybe if we put the chromatin in a more open state, this could help the regeneration process. So Nick used a drug called TSA, which is a histone deacetylase inhibitor. In addition to overexpressing ASCL1, and by the same method I told you about before, we used the GLAST Cree that drives TTA, and then that will activate the TETO ASCL1, and that will also generate a GFP to allow us to trace the cells. So again, here's what normal glia look like when he added ASCL1 to these glia, as well as injected intravitreally a TSA. He found that now, even in adult mice, we started to see some neurons appearing. And so these were examples of amacrine-like cells and some examples of bipolar cells. And so morphologically, this was really very much like what we saw in the young mice. These cells didn't take on perfect morphologies of the types of bipolars or amacrines, but they were reasonable enough that we could recognize them in their morphologies. And I'll get to this later in the talk, that now we know there's many different subtypes of bipolars that are produced, not just some generic one. With Rachel Wong's group, we analyzed synaptic connectivity. We could see that these cells were making close opposition to other cells that had ribbon synapses, as well as in the interplexiform layer, we could see examples of opposition between, say, CTBP2 and PSD95, all within the process of one of these cells. And also with Rachel's group, we did serial block-based EM reconstructions. And so we could find, using this method where you identify with 2-photon where your cell is, the potential neuron in a flat mount of the retina, and then you crank up the laser to burn a little box around it with a couple little arrows pointing to the cell, which is right here. And then you do serial block-based EM reconstructions, and you can find that cell and reconstruct it and show, in this case, that a cone was synapsing with this process of the Mueller-glial-derived neuron. And then with Fred Ricci, he made retinal slices of some of these cells, and he could show that glial cell-derived neurons, in this case a bipolar-like cell, if he patched onto it, he could get a light response that was very similar to what you would see in a normal bipolar cell. The normal bipolar cell always had larger potentials than the Mueller-glial-derived bipolar cells, but in general, many of these would show light responses, and we can see that here as well. You could both see on and off responses in these cells. So again, we knew we were having a diversity of bipolar cell type. At that time, we also did some initial single-cell RNA-seq, and I'm going to show you more of this in a few minutes. But the Mueller-glia form one big group from the single-cell RNA-seq data, but these progenitor-like cells form a distinct group from the Mueller-glia. And these cells are ones that have ASCL1 turned on in them, but they seem to stay in this progenitor-like state. But then a group of cells with ASCL1 turned on in them, they become bipolar and amacrine cells. And so in all cases, what we did for this was we fax purified the green cells, so cells that were lineage traced. So what we're seeing here are really just lineage traced cells. But I have to say, because of the number of rods in the retina, back in some of those early stages, we would always see a small cluster of rods as well. And we interpret these as being rod contamination in the single cell data set. But we can come back to that in the question period as to whether we're actually seeing any rods, even in those original studies, or if those were not glial derived. one way we could tell that these cells were glial derived is they retain some glial gene expression. So they don't lose all their glial gene expression. And I have a feeling that that's why they don't look exactly like normal bipolar cells, is that they've carried with them some of the glial phenotype. Eventually that does get silenced though. In the ATAC-seq data that we did in this paper, we could see that the glial gene program is getting silenced even at this stage. Yeah, that's shown here. You can see like accessibility at the RL. So this is a DNA accessibility now done with ATAC-seq, but you can see right around the RLBP gene, which is the glial cell specific promoter here. You can see that goes away with ASCL1 or with the ASCL1, NMDA, and TSA, which we call the ANT treatment. The glial gene expression goes down, and neural genes, then accessibility comes back up. So our conclusions up to this point were that epigenetic factors limit the regeneration in the adult mouse. But if you combine ASCL1 with HDAC inhibition, this allows the epigenome of the glia to be remodeled to allow regeneration, and that then these regenerated neurons wire into the existing circuitry and respond to light. Now, since these studies were done, we've pursued a number of different directions to try and make this better. How can we improve it? We're seeing bipolar cells, some amacrine cells. We're not seeing great evidence for photoreceptors, and we're not seeing much of the way of ganglion cells. And also it was really a relatively small percentage of the cells that actually end up going all the way to a definitive neural phenotype. So we've been playing around with both intrinsic and extrinsic factors to try and see if we can improve the rate of reprogramming, and number one. And number two, or modulate the types of neurons that get regenerated. And that's what I'll tell you about now. So one of the things that we did to look to see if we went back to our epigenome to ask the question, ASCL1 is driving this process, so it must be bound to these sites in the Mueller glia of the mouse that are allowing it to drive gene expression. But in addition to binding to its own site, to the sites that it should be binding to, is it binding to inappropriate sites, or is it limited even with the TSA in the sites that it can bind to? So we did ASCL1 ChIP-seq, which basically involved just doing normal ChIP-seq on developing retina, where ASCL1 is only in progenitors, and then comparing that with overexpressed with the ChIP-seq that we would see from ASCL1 when we looked at Mueller glia that are overexpressing it. The developmental ASCL1 of retinal progenitors showed us that when we compared it to neural progenitors or spinal cord or retina, when we combined the three data sets from ChIP-seq, we saw that there was a sort of a core neural progenitor set of binding sites for ASCL1 that drives all the usual suspects in neurogenesis, like deltas and NFIs and Notch, RBPJ, SOX11, SOX2. So all these key progenitor genes are being driven by this core set of elements. And you can see ChIP-seq data for some of those elements here. In addition, there are retina-specific regions, and these things are near genes like 807, DKK, ID1, PAX6, OTX2. And so the retina-specific regions are not shared in neuroprogenitors or spinal cords, but these tend to be really important for making more retinal-like neurons. And so I think using the data from the ChIP-seq allowed us to sort of separate into the sites that were more retina-specific versus more neurogeneric. Now, one of the things when we compared them to glia, here's the ASCL1 overexpressing glia that are being reprogrammed, and you can see some of these sites are the same as what we see in the neural progenitors. So these are the good ones. These are the ones we want. But then again, sometimes you see sites that are in areas that the progenitors don't have. And these are inappropriate sites that presumably were open in the glia and have an E-box, so could bind ASCL1, but we call them ectopic sites or inappropriate sites. What we found is that Mueller glia have a ton of these ectopic sites. And so P0 retina these are the sites in the normal that we would call appropriate. And this is the overlap between the progenitor sites that bind ASCL1 and the Mueller glial sites that bind ASCL1. And so you can see there's lots of overlap. We haven't got every site engaged yet that the P0 retina has. So there's still some missing here, but there's a lot of sites here that ASCL1 is being bound to that are presumably not appropriate, not driving developmental gene expression like we would like. And when we looked at what motifs were represented there, we found actually that one motif in these sites that's not present in these sites is a STAT binding site. And so 12% of these regions had STAT binding sites in them. And some of these were actually at places when we did chip, or when we analyzed a chip seq for STAT, some of these were places that drove genes that actually inhibit neural differentiation, like ID3 and ID1. And so these sites that we found in the glia that could drive these things, we thought might be inhibiting the process of regeneration. So we went back to our ASCL1 and TSA paradigm, but now we included STAT inhibition. We were thinking that since after injury, STAT signaling really is known to increase in Mueller glia, could this be sort of inhibiting or pushing back on our regeneration process? And so we now combine TSA with a STAT inhibitor and with an intraocular injection. And now what we found is much better regeneration. So now you can see in this field, there's hardly any normal looking Mueller glia, and now everybody starts to look like a neuron. And in this case, we can also see some cones. These are not Mueller glia derived cones, but we just looked for opposition between cone pedicles and some of these new bipolar cells that we were generating. And you can see really nice opposition in some of these cases. In addition, we hadn't really analyzed in the previous paper whether these Mueller glia re-enter the cell cycle after injury. And we've also, now that we had more improved stimulation of regeneration from the Mueller glia, we actually looked to see if we could see EDU incorporation into these glia. And we see lots of EDU incorporation into the glia after damage and with this new paradigm of inhibition of STAT and with the TSA. And so you can see lots of cells here are EDU positive, and they're also labeled for CABP5 and other neural markers. So we see a big increase from ANT with the ANT STAT inhibitor signaling in terms of the number of neurons. We also went back and looked at where ASCL1 was bound, and we found that there was a big increase in binding of ASCL1 now to neuroprogenitor genes, and a decrease in genes like ID3 and HES15, where MASH was no longer binding to these inhibitory sites. And you can just see here, this is just quantifying the total number of glia versus neurons. This is the wild type, it's all glia. With ANT, you can see that there's definitely some neurons coming in and progenitor-like cells, but with ANT-C, this is with the STAT inhibitor as well, now we see a big increase in the number of neurons, definitive neurons that are generated by this treatment. So now we have this improved by going back to the epigenome and actually trying to do more targeted epigenomic reprogramming by antagonizing this STAT signal. I think we've really improved the regeneration process a bit more. And this was published, Levi and Nick Jorstad got together on this one to really drive this one through, and it was published in Cell Reports last summer. So Levi's going to talk more about this, I think, in an upcoming NEI talk. Now, I just want to close with a little bit more about additional transcription factors. We've been testing a wide variety of additional transcription factors because our thinking is that if we put in ASCL1, this can help bring the cells to a progenitor-like state. But now if we add additional transcription factors to that mix, will we target to a particular cell type? One of the transcription factors we were using 80, in this case 801, but it's similar to 807, ADO transcription factors we thought might improve our yield of retinal ganglion cells because ADO7 is necessary during normal development for retinal ganglion cell regeneration. So we combined an ASCL1 mouse with the 807 mouse, actually 801 mouse from Ping Chen, and we put these mice together, and now we're driving these two proneural BHLH transcription factors. And now we saw something really remarkable. Now without adding the TSA, without adding the statin inhibitor, and actually without even damage, you can convert Mueller glia at an extremely high efficiency into retinal neurons. And so now we can look over here, we can see normally we would see neurons occupying this bit right here with Mueller glia here in the ant treatment. Now all these guys are neurons, and it's up to 80% of the cells now become neurons. And they take on a variety of different morphologies. Again, we start to see cells now in the outer nuclear layer that look a lot like photoreceptors. And when we do single cell RNA-seq on these guys, we can see there's practically no Mueller glia left. It's just a tiny little group of Mueller glia now left. There's a big group of progenitor cells now, but then there's most cells are pushed right on through the progenitor state into this immature neuron state, into bipolar cells and amacrine cells, and now we see cone photoreceptors arising from the glia. And we can see this with things like opsin, as well as other cone markers, restins, the opsins, both short wavelength and middle wavelength opsins are coming on in these cells. And so we're really excited about this new combination of transcription factors that seems to just push the glia right on through. We don't really have a molecular explanation for this yet, and we're now going on doing ChIP-seq for ADOs and ASCL1 to look whether that combination or even heterodimer formation is what makes this just much more efficient. But it's really pretty impressive. And just show you a couple other things that I just sort of hot off the presses kind of data. But you can see that the progenitor cells are sitting here. They're expressing deltas, IDs, Notch, S6. But now this group is also expressing these immature neural markers like TBX, NeuroD2, NeuroD6, EBF1. These are all markers of maturing neurons. And so we push cells right on through from Mueller glia into this immature neuron state. And then now they really, like the bipolar cells, make pretty much every marker for bipolar cells that comes out of the recent Josh Saines' papers about bipolar cell diversity in the mouse retina. You can see those markers in populations of these bipolar cells. So it's a wide variety of different bipolar cells, and as I said, also a group of cones. And this has really been only after a couple weeks. So now we're waiting on the data set for letting this go for 6 and 8 weeks, like what Nick had done. So I think we're really seeing that you can manipulate the phenotype of the glia quite dramatically. And so I think that, I'll just wrap up by saying I think a combination of epigenetic modulators and different transcription factors, as well as some of these things like manipulating the STAT pathway, are all showing us what the limits to these in vivo reprogramming approaches can be. And I think we still are having lots of surprises as to how to make this more effective and how to actually get better control over the types of neurons that we make. But one of the things that really gratified me is that it's been really hard. I've done a lot of transplant work as well, and embryonic stem cell transplants, and I think it's been really hard. In fact, it's a problem that the whole community is grappling with, is how do we get those neurons from the subretinal space into the retina, and how do we get cells from the vitreous into the retina? And transplantation is always going to have this limit that the retina is a very thin piece of tissue, and it's going to take a really good surgeon or figuring out what these barriers are to transplantation. But I think with this approach, you're actually remaking neurons inside the retina, and we can see that once you do that, whether it be the bipolar cells or amacrine-like cells, and Fred's now recording from these new mice, so we soon should have more data about whether photoreceptors will do the same thing. But I think what we can see is that once you put these cells back into the retina circuit inside the retina from within, regenerating from within instead of without, then they do make synaptic connections. So the available machinery is present in the mature retina to let cells wire back in, even in adults, at least in mice. So I think, to me, because I'm sure there'll be other problems with how to move these techniques into clinical therapies, but I think they are not encumbered by some of the more thorny issues that the transplant field is currently running up against. So yeah, I'm optimistic, and I also think that I think we'll build out from here all the different things that affect this reprogramming process, both intrinsic and extrinsic, and hopefully we'll get better control over the entire process and really realize this as an ultimate therapy for repair. With that, I'm going to close and thank my lab. They've been great. I talked about a lot of work that was done by Nick Jorstad. Nick was a grad student in the lab, and really none of this would have happened without him. But I could say the same. Yumi was really early, was one of the prime movers of this early on, and must have crossed 15 different mice until we finally got the combination that worked. As well as more recently, Levi, who's been really spearheading the additional efforts along with Marcus Hooper and Wes Jenkins to find additional transcription factors to reprogram to diverse cell types. And I want to thank the Eye Institute, the Foundation Fighting Blindness, the Gilbert Foundation, Open Philanthropy, and Genentech, my funders. And thank you all for your attention. I'll take any questions.
Thank you so much for a fantastic talk. We have about 10 or 15 minutes for questions. You guys can post them in the Ask a Question or the chat box. And to start off, we have one question, is there a limit on Mueller glial cell regeneration? For example, if there was complete degeneration of the ONL, how much can we regenerate with Mueller cells?
Yeah, I think that's probably, that's a really important question. We see division, we see cell division. I think the ASCL1 puts the cells into a proliferative state. It certainly turns on things like cyclin Ds, et cetera. So we know that also from Nick's paper, if you stimulate that, if you add a growth factor like EGF or FGF through an intraocular injection, you could stimulate the proliferation even more in these ASCL1 expressing glia. So I think in theory, you could make a lot of cells, but in practice, I think that's going to be a very hard thing to regulate. And certainly when you think of the hundreds of millions of rods we have in our retina, it would not be my first target to go after a rod degeneration. I think when you think of cones, it's a more manageable process because the number of Mueller glia, and like in the fovea, for example, and the cones is a little better matched, and you may not need to regenerate, all of the cones to get function, some recovery of vision in someone who didn't have many. So I would think cone diseases, maybe, if people like, Jeff Goldberg can figure out how to regenerate the optic nerve, maybe ganglion cells is a reasonable target for. I know central retinal arterial occlusion. There's evidence that there's sometimes ganglion cell sparing, but more inner retina damage, like amacrines and bipolars. And if that's true, then in those cases, then we can certainly make a lot of those cells. But I think there are definitely limits to the types of diseases you could potentially treat. But yeah, other than rods, though, I think the numbers could work out, because if you can get a few rounds of division out of these cells, or even direct transdifferentiation, which we're also pretty sure happens in some of these cells, we think that even that could help in cases where you've lost all your neurons but still have plenty of glia left.
And then along those lines, it looks like there's two related questions. On the flip side of the coin, if you're able to regenerate, if almost 80% or more of the glia are turning into other cell types, what's the consequence of not having those glia in a retina anymore?
Yeah, I think that's, again, a really important question. one of the things that bothered me about the first, when we only had ASCL1 alone on board, was that, in these mice, you only get recombination of about 10% of the glia. So what you were seeing in all those images was only about 10% of the glia. And also the Glaskria and the ROBP-Cria are kind of spotty, so often you'd see just a patch of glia or whatever. I mean, obviously, if you were thinking about this for a patient with a gene therapy approach, you'd probably want to titer it so you were infecting only a subpopulation of the glia. Because I think, if you hit all the glia, I think you would definitely have some issues. So the ideal would be to, try and titer it, so you dose it so that you only hit a subpopulation. But having said that, knowing that 80% of those are going to switch, that gives you a lot more control, because 80% of the ones you hit will switch. And so then at least you can feel good that your equivalent dose is going to be roughly, what you see is kind of what you get. Whereas if only 10% of them switch, you have to infect a lot more than what's ultimately going to switch into a neuron or give rise, regenerate neurons. Then I think it's a little trickier. But again, these are really good questions. I think it really, when it comes to a therapy, we're not even close to that.
And then another question, you mentioned Dr. Goldberg's work. Do you see ganglion cells that are being differentiated from these glia?
Not with this combination of transcription factors, but with another combination of transcription factors, we do. And so I think that it's definitely possible to get ganglion cells. They're not, robust, healthy, happy, perfect ganglion cells by any stretch of the imagination. And Fred's recorded from some of these, and they make little mini spikes, and they make little wandering axons that don't really go the right way most of the time. So I think, again, we can make some cells that are thinking about being a ganglion cell when they grow up. But so I think it's possible. I don't think there's an intrinsic limit against making a ganglion cell from a glia. A couple of years ago, we just weren't sure if there were somehow basic limits to what the glia could do, because these are sort of evolutionarily old limits. It seems like if the bird already has restriction on what it can do from what the fish does, you sort of have to wonder. But I think these transcription factors are powerful. They do stuff to cells to make them change their program of gene expression, and we've already seen that sometimes those things they're doing are not the right things. But if you can refine the approach to where you just activate the right things at the right time in these cells, I think that you can ultimately hopefully make these neurons more perfect. I think right now they're not perfect. I think we have a ways to go, even on what the phenotype of the neurons are that get regenerated.
I found interesting in one of the last single cell data sets you showed with the ASCL1 and ATO1 that you had a small population of cones, but not rods. And I wonder if you could speculate on that, since I would have expected rods to be much more similar to bipolar cells and Mueller glia, given that they differentiate naturally at a different kind of time than the cones do.
Yeah, and part of that I think might be what 807 or 801 do. I think they are expressed early in retinal development, around the time that ganglion cells and cones are being made. And we know that if you knock out 807, you lose ganglion cells, so you lose this early fate. And it does seem like ASCL1 can replace 807's role, at least in part, but it also seems like there's a different set of genes that are turned on by 801 or 807 that are I guess you could call them early progenitor genes or early postmitotic progenitor genes on their way to these early fates. So yeah, it was surprising. Like I said, I think we just don't really know exactly how best to push these things around. But we've tried a lot of the typical transcription factors for cones. We tried, even when we put in ASCL1, OTX2 gets turned on in virtually every cell. So because they make mostly bipolars, we thought PRDM1 blimp, which Joe Brzezinski showed in my lab, is necessary to toggle between bipolars and photoreceptors, we thought it was a no-brainer to just overexpress BLIMP and get all these bipolars to switch into photoreceptors, but that didn't work at all. So most of the predictions that we make still are pretty far off the mark, and we're just trying to kind of test a lot of stuff, and hopefully some of it will give us cell types that are useful.
Another question that we have was, what mechanistic impact does injury have, both positive and negative, on regeneration of neurons in mice that express ASCL1 and Glia?
So the injury we thought was really going to be necessary, but it was necessary if we just have ASCL1 in the mix. So we thought that possibly, like in the fish, it's known that the injury and the STAT signaling that follows the injury is important for actually getting the ball rolling. So if you block STAT signaling in fish glia, they don't regenerate. David Hyatt's group has shown that. So it was interesting. We were sort of surprised, but on the other hand, it does look like one of the things that's different in fish versus mice is in mice, the STAT signal stays on for at least a week or two, and in fish it's transient. It goes up, activates. A lot of these genes like IDs get driven partially by STAT signaling and BMP signaling, and then that goes down, and the IDs then no longer repress the proneurals. And so the proneurals can kick into action and make the neurons. But during that initial phase where IDs 1 and 3 and Has5 and all Has1 are driven by these alternate signals like STAT, the progenitors get their start and can make more cells. So it could be something like this, that if we could balance it right, an initial injury would still be a good thing, but then, get rid of it, which is, I think, what we're doing with the STAT signaling. I should add, Levi had a paper just in December on microglia, and we think that maybe at least some of the STAT signaling may involve the microglia, since getting rid of the microglia with Plexicon gives you the same effect. It essentially looks a lot like what I showed with the inhibition of STAT signaling. So it's quite likely that after injury, you get this microglial response that activates STAT signaling both in the microglia and in the Mueller glia. Lots of cytokines are released that are great for the injury response in fish, but in mice, because they tend to be persistent for much longer, actually to inhibit the process.
And can you comment on the immune system in terms of the microglia interaction with these processes? And do you think that they would be very different in a disease state where the immune surveillance and barriers are quite different than they are in a normal functioning retina?
Yeah, and I think that's where like this acute injury, like ASCL1, like NMDA, versus a more chronic injury, like a P23H or something, where it's a slow degeneration process. I'm not really sure. With ASCL1 alone, Nick Jorstad did do the following. He did this experiment. We never published it, but it's interesting. He crossed these guys onto a Pro23 his H mouse, so he could elicit degeneration in the retina of the rods. And in addition, he did a light damage, and the light damage did cause an initiation of this regenerative response. So with ASCL1 and light damage, we did see new neurons being made. They weren't rods, they were bipolars. So kind of weird. So I think it's a more generic signal that, the damage caused. And light damage will give you pretty much the same result as NMDA. But what's interesting is the slower degeneration of the rods didn't induce this effect. So I don't know if the microglial response is different in some slow degeneration like a rod disease than it is from an acute injury like an NMDA. But it might be that there is a difference there. Levi's just really looking into this now. All the other immune cells that enter the retina after these injuries and how they interact with the microglia, and it's going to be really interesting, I think. And there will, this is probably at least one thing that differs in fish versus mice, is that the response to injury and the immune component and the way the immune cells interact with the glia probably underlies at least part of the explanation for why we don't regenerate.
Yeah, that's very interesting. I think we have time for maybe one or maybe two more questions. One is, there were a couple of high-profile papers last year showing that PTBP1 knockdown can convert glia to neurons. Have you tried PTBP1 knockdown in the glia?
So we did something very similar in vitro. Stephanie Wohl did these experiments because we were looking at microglia, MIR124, and CTDSP1 is also part of that PTBP1 pathway. And so she found that microRNA 124 overexpression, MIR124 overexpression, or dominant negative CTDSP1, which also, and as we monitored it, we could see PTBP1 changing in the way that was predicted here, where you got an increase in it in the glia. And indeed, that does convert these glia into neurons with some efficiency in our hands as well in the cultures. What it does is it turns on ASCL1. So it's upstream of ASCL1, that pathway. Now, we have never done that in vivo. And in that study, it has been criticized that some of the AAVs that they were using also infect these other neurons. So I think you have to be really careful in interpreting some of this work that's coming out about reprogramming glia to be sure that in our hands, the reagents are pretty well validated. I mean, extremely well validated, because we've been working with them for so long, and it's transgenic mouse based. And so, you can look at one mouse, the next mouse, the next mouse, and have a lot of reliability. I think with some of the AAV work that originally characterized SH10 as being a glial-specific capsid for AAV, it's coming out that if high enough titer with a subretinal injection, you have a lot of photoreceptors with that. So I think we have to be pretty careful about using capsid-based approaches to confine expression to particular cell populations. But obviously, going forward, we're going to need to really nail this down, because we're not, all the next translational steps aren't going to be using mice. So
right. Okay. Well,
get it. Yeah. Okay.
No, with that, I think we're out of time, so I will close it there. But thank you so much for joining us today. It's really a fantastic talk and really exciting work.
Okay, great. Well, good to talk to everybody. All right. And, bye-bye.
Bye.